Okay, so I'll go straight into it. We've got three people talking today. We've got Pat Hayes, who most of you I think on the call know. Pat's still a professor at the University of Oregon. You'll know that he's a geneticist and a plant breeder who's been trying to develop varieties that meet end user needs, particularly in relation to malting and brewing, but also for food and for feed. I read Pat's little blurb before I came on here, and I saw that, you know, in the period of time he's been at Oregon State University, the Barley Project has released 18 varieties, developed and shared 21 mapping panels, distributed 20 metric tons of barley seed, published 168 papers, authored 13 book chapters, and taught plant genetics to everybody, I think, that's a plant geneticist in the United States. So today, Pat's going to be joined with two what I'll call early career scientists. I hope that's not an offensive term. That he wants to introduce to our scientific community. The first of these is Chris Massman. Chris works with Lucia Gutierrez in the University of Wisconsin. He's a PhD student in plant breeding and genetics and is working mainly on naked barley. The second person is Helen Brabham or Brabham. Helen, can you tell me what's right? Brabham?
Yeah, Brabham, yes.
So Helen works in the Two Blades group. She's a team leader in the Two Blades group at the Sainsbury lab in Norwich, and she's been working with these guys, as far as I understand, doing some genetic engineering basically with some of the targets that they've been working on. So I'm going to pass you over to Pat soon, who's going to compare this multi-partner presentation. The topic of the talk or the presentations is barley of the future, facultative, naked, and flavourful, and that's a provocative set of topics that I will anticipate will generate lots of questions and comments. So my last comment is that if you want to ask a question, please pop it down in the chat box. You can open the chat box, I think, in the app, and then we'll come back to them at the end of the session. Please note who you're asking the question to, though. So Pat, over to you.
All right, thank you, Robbie. Let me get the screen sharing going here, and a thumbs up if you're seeing the full screen version. All right. So thanks, Robbie, for the intro. And just a quick update there is that Chris Massman is finishing up his PhD, and we'll do that next week, I believe. And then we'll join the Barley Project as a postdoc. He's officially part of the Barley World team as of next week, or May 1 is his official start date. Helen has been a long-term cooperator in a number of ventures, and in addition to doing the gene editing work that she'll talk about today, we've been collaborating on stripe rust resistance genes for some time. More broadly, I'd like to thank the International Barley Hub for inviting me to this seminar. Their effort to re-galvanize the barley community is most welcome, and it's in that spirit that we invite some of our younger scientists like Chris and Helen to join the talk. And hopefully the title isn't just provocative, but is intended to get this new generation thinking that that could be pretty cool, or no, that is definitely something that we don't want to do. So. What do we mean by the future here? So crystal balls are always a bit challenging, and thankfully the CSA News, this is the Crop Science Society of America, an August journal came out with this cover here, and if you look closely, it looks to me like this artist rendition is saying that we're going to be raising barley and hops on Mars. So I think that would suggest that there is definitely a future for barley in one form or the other. But to move into some definitions of what we'll really be focusing on today in terms of what we mean by facultative, what we mean by naked, and what we mean by flavorful. Facultative is at this point just simply defined as barleys that will flower in a timely fashion without vernalization. And so here we're looking at a field planting of some various accessions in our program. This is planted in the spring, and if this row flowers in a timely fashion, then we would say, hey, that has the potential to be facultative. And these that are not flowering in a timely fashion are going to be extremely sensitive to vernalization, and so they do not have a hope of being facultative. Now, there's some additional caveats in the definition of what's a facultative, because not only do you need to flower in a timely fashion without vernalization, but you need to have the full, complete, full-bore low temperature tolerance regulon. So you have to have a plant that is capable of acclimating and achieving maximum low temperature tolerance, but at the same time does not require vernalization. We'll get into that a little bit more as to why facultative growth habit is, I think, a tool that we can use to address some of the challenges of climate change. Naked is up here in the right panel. And that naked is just such a handy and botanically correct term, referring to the adherence or non-adherence of the lemma impalia to the Caryopsis. And you can, the other terms are hull-less and so forth, but that gets really grammatically complicated to go there. So we just follow the example of the group that initially cloned the gene and call it naked barley. These are naked barleys in the center of the figure here. You can see all sorts of colors. Once the hull is gone, then you're able to see a lot more of what is going on in terms of the kernel color. The exterior, the border of this is pearl barley. And so that's barley that has been mechanically abraded in order to remove the hull so that we as humans can eat it, because the hull isn't particularly tasty. It's going to be insoluble dietary fiber. Flavor, we're showing down here in the lower panel, and this was one of the bits of just heavy lifting that we have to do as barley researchers, and particularly once we get into sensory, and that is that you are required to taste beers during presentations. And in this particular case, this is a lineup of beers that were part of one of our flavor projects at a Master Brewers Association. And flavor is just this incredibly complex phenotype that we're just starting to get a bit of a grip on. And we've got an evolving phenotyping platform to deal with that. To date, our efforts have been really directed at beer, but there's some kind of things on the horizon in terms of spirits and in terms of roasted barley tea as well. So let's dig into this facultative growth habit a bit more in terms of its definitions. And so some key overall considerations are low temperature tolerance, fertilization sensitivity, and photoperiod sensitivity. Going into the weeds here just a bit, that we're referring specifically to sensitivity to short days, and that is that the plant will not transition from a vegetative to a reproductive stage until a sufficiently long photoperiod is achieved, or in other words, under short photoperiods, the transition will not occur. So in winter growth habit types, yes, they should be low temperature tolerant if they're being planted in the fall. In the Northern Hemisphere, yes, they are vernalization sensitive. And interestingly, their photoperiod sensitivity varies, their short day sensitivity. So as we started digging into germplasm collections, we've seen that some winter barleys are in fact not sensitive to short days. They are relying only on their vernalization sensitivity to remain vegetative. And I would argue that that's a dangerous insurance policy during climate change. So facultatives then, yes, they are resistant to low temperature tolerance. They are not sensitive to vernalization. They do not require vernalization. And yes, they really need to be sensitive to short days, because otherwise there are no brakes to put on the transition from vegetative to reproductive transition. Spring types, why, no, they do not have low temperature tolerance. No, they are not sensitivity to vernalization. And the photoperiod sensitivity varies curiously. Every once in a while in a germplasm survey, you find out that there are short day sensitivity alleles in what we would call spring growth habit barleys. But that gives us kind of a set of gross definitions to move ahead then. So why do we care about growth habit? Well, because of climate change. Plants are not able to do what we're able to do as humans, which is just to get up and move in response to climate change. But instead, they're required to remain stationary and, as in the case of this tree, swallow the thermometer that is in fact diagnostic of the fact that, yes, climate change is indeed occurring. But, why you may be thinking, wait a second, low temperature tolerance in the context of of climate change, which is net warming, we are experiencing increased volatility. So there are still low temperature events. So we need to be bearing that in mind. Drought is a feature of climate change. We're seeing new diseases in familiar places, familiar diseases in new places, and just so our life is being more complicated there. And in the face of all this, we need to maintain productivity and quality. I've got a hyperlink down here that is an interview that I did with a group called the Ag Climate Network at Washington State University. And so they've got some additional resources there on barley breeding and climate change, if you'd like to go there. But, really what's at the crux of all this is spring versus fall planting. This is an aerial view of some of our research plots here in Corvallis, Oregon. This is fall planted barley in the foreground. And a number of years ago, Robbie saw a picture of our fall plots and asked if that was miscanthus. No, in fact, that's fall planted barley, and it's incredibly vigorous, and it's going to yield about 25% more than this spring barley in the background. And furthermore, note that this spring barley is being irrigated. So we are stealing water, a very precious resource, from salmon and people and other higher value crops. So fall planted barley, I think, is really the way to go. But if you're going to fall plant your barley in many areas, you're going to have some low temperature tolerance, and low temperature tolerance is an area of endeavor that has just a noble history of physiology and genetics research. We know it's an acclimation trait that you have to have a sufficient exposure to low temperature to trigger the regulon. It is lost. Whatever low temperature tolerance you have is going to be lost when you affect that vegetative to reproductive transition in terms of the vegetative stage cold tolerance that we're talking about. There's a whole other area of endeavor, which is the reproductive stage cold tolerance. That would be, for example, the fitness of the pollen in the egg to survive frost at anthesis. And that's really important to some of our colleagues in Australia, but our focus is on the vegetative stage low temperature tolerance. So you got a couple of ways to put the brakes on that vegetative to reproductive transition. One of them is vernalization sensitivity, and the other is the short day photoperiod sensitivity. And I would argue that if you have vernalization sensitivity only, then your transition may occur in a premature fashion. If you get enough warm units by February in very dangerous locales, you can transition and then be hammered by a low temperature event. If you are held back, in contrast, by short day, that's not going to change with climate change. Day length, one hopes will not change, then you may be able to regulate that with the sensitivity to short days. So all that said then, why facultative growth habit? It provides maximum flexibility to growers, to industry. This is my colleague, Scott Fisk, looking at a production field of our variety Lightning on farm here in Oregon. And Lightning is a facultative barley that yields quite well in our environments, fall planted, and can generally defend itself fairly well in a spring planting as well. So in a fall planting, then you've got cold tolerance. It's there if you need it. And in spring plantings, the same variety can be planted, but the cold tolerance regulon is not needed, and therefore it doesn't need to be invoked. So you can then think about multiple uses of your facultative barley, that it could go into beer, could go into food, could go into feed, and we'll talk a bit more about multi-use when we get to naked barleys. Right now, I just wanted to briefly recap a paper that we published recently with Maria Munoz Amatriain as the lead on it here, and then a lineup of just outstanding barley researchers who helped to contribute to this research. So it was this was one of the last bits to come out of the Tritici ECAP initiative, and so we call it the TCAP LTT project. And it was a rather ambitious endeavor that involved a large GWAS panel contributed by breeding programs, genetics programs around the world. We had 2 years of phenotyping. The Illumina 9K was available at that point. We had some CASP allele-specific genotyping thanks to Lima grain. We measured low temperature tolerances, winter survival in these field environments, and vernalization sensitivity as days to flowering without vernalization. And as you see in these histograms here, in terms of percent survival, we have a a very nice kind of normal distribution. And we see that full pint, our spring check, is down here with the dead. And Alba and Maha are winter. Alba is the winter check. Maha is the facultative check, are co-performing with the higher survival group. There are some transgressive segregants, if you will, out here. So those are of particular interest to us because what do they have that is giving them the extra bit of low temperature tolerance? Days to flowering, on the other hand, so we see that full pint, the spring, and Maha, the facultative, are flowering at the same time. And we see that Alba is over here with the late flowering group. And if this wasn't a GWAS panel, you'd be tempted to say that that was a 3-to-1 ratio, but I think it just shows that the preponderance of winter types that we had in this collection. So digging in a little bit more here, just to kind of characterize this germplasm collection a bit, if we look at the top and bottom 5% for survival, we see that the full pint is over here with the kind of low surviving group. We see the higher surviving group over primarily up in this upper right quadrant with Maha and Alba plugged right in here, and then a fair number of interesting individuals down here. We move over to row type, and alas and alack, we see all kinds of 6 rows down here, and then the 2 rows are sort of clustering out here in the middle and then over into the left with the springs. But do not despair, that was just kind of ascertainment bias because we had a whole lot of 6 rows in our collection. That's what we were working on at that time. And then likewise with vernalization, we classified the types as being intermediate, sensitive, and insensitive. The unsensitive would be the facultative types. Note that we have a lot of intermediate and sensitives over here in the high survival group, but there are a sprinkling of facultatives. And again, I would say that's probably an ascertainment bias problem because most breeding programs have not been consciously thinking about facultative growth habits. So there were relatively few of them in the program. So then we look at our Manhattan plots here, and we just dug up a couple of suspects, old friends in this process. And so we see that in terms of days to flowering, that'd be vernalization sensitivity. Just our monster hit is Vern2, which is just a key driver of vernalization sensitivity. We see Vern1, also important, PPDH1. Now this is H1. This is sensitivity to long days is showing up down here. And we're not seeing PPDH2. And you may wonder why, and I'll show a bit of data on that in just a second. Let me explain why we're not seeing PPDH2. When it comes to winter survival, then just going left to right, here we see on 1H a friend FRH3, which is something that our colleague Scott Fisk discovered as a QTL during his master's thesis research here at OSU. And then moving across, we see Vern2 emerging as an important determinant. I would argue not because of vernalization sensitivity, but just because of correlation, not causation, that many of our low temperature tolerant lines, in fact, are sensitive to vernalization. FRH2, which is just a famous collection of low temperature tolerance genes. These are CBF transcription factors and a multi-gene family, just the huge driver here. And then FRH1, aka VernH1, also showing up as an important determinant. So if we look just a bit more at some of the allele-specific genotyping that at this time we did with thanks to Lima grain, and that then now Javier Hernandez has developed a set of Cas markers which are now available at the Barley World site if you'd like to pick those up for these loci. But we can look at kind of just some key differences between the highest surviving accessions in the panel here. So this is the winter survival in the first column, days to flowering in the second column. So these first set up here from 60 to 69, these would be individuals that we would describe as potentially facultative because they are flowering right along with the springs. These are the winters because they're flowering in a very delayed fashion. It's interesting to see what's happening with PPDH2. So this is the short day sensitivity in these individuals. The loss of function is the short day sensitivity allele. And so we see that here in these facultative individuals, we have the loss of function or the short day sensitivity allele. And then here in this individual from Minnesota, we see that it in fact has the intact allele. And I would argue that that's a bit dangerous for a facultative type because you might transition prematurely. And then we see a sprinkling of either functional or non-functional PPDH2s. And then the succession from Russia down here is just a curious one for all of the allele types, and something that certainly bears additional inspection. And our colleague Brian Stephenson and colleagues have been looking at another panel of barleys from the Vavilov collection relating to low temperature tolerance. So PPDH1, fascinating in that we see that our fall planted barleys, facultative and winter, have the functional allele here, whereas spring barleys do not. So here's an opportunity. If you wanted to increase the yield of fall planted barley, you could think about a substitution there. All of these have the intact intron 1, which has been identified as a potential determinant of low temperature tolerance. And then in VERN2, we see a deletion of either 2 or more of the gene family members that comprise VERN2 in all of the facultatives. And then as we move into the winters, then we see that there are some partial deletions, and here a deletion of the HC version that may be diagnostic of who are the key drivers here. And so it looks like kind of A and B, and this has been shown up in some other research as well. So then we can just kind of look at some of these groupings that we see here in terms of the top 5%. Where's this germplasm coming from, and what sort of attributes does it have? So there's a group up here that was comprised of primarily Minnesota lines from Kevin Smith's program. And Kevin is embarked on a very ambitious program of selection for low temperature tolerance in an environment where that is key. These are selections from our program. At the time, we were working on 6 rows. These were all 6 rows. There's kind of 2 groups comprised of some European germplasm here. Then we move over to Nebraska. There's kind of a unique cluster here, and then a lot of Nebraska material up here as well, with a sprinkling of lines from Virginia Tech. And then there's this group of PI lines that originated in Russian collections. So some sort of next steps with this panel are that we'd like to further explore facultative growth habit. We'd like to isolate FRH3, find out exactly who that is. And this whole panel has been transferred then to Tim Bernard and the group there at University of Gießen. So they'll be looking after that for dispersal throughout Europe if you're interested in accessing that collection. So what are some prospects for facultative growth habit? This is a shot of our fall planted breeding program here in Corvallis. And so we're quite keen on promoting facultative growth habit, but we continue to release winter varieties. In our experience, really, the marker-assisted characterization is very useful, but there is no substitute for phenotyping. And that is that you just plant your material out in the spring and see if it flowers or not. Another approach to actually getting towards agronomic competitiveness with facultative types is going to be to then really run 2 breeding programs. So you would have your fall planted breeding program, you'd have your spring planted breeding program, and then you may require additional environments to be able to actually measure low temperature tolerance. Here is just a characterization of lightning using the cast markers that Javier Hernandez has developed. So I'm quite optimistic that we'll be able to move onwards towards the facultative growth habit in a range of different barleys. So now we're going to turn our attention to the second term in our title, which was naked. And so we're going to talk a bit about naked barleys, and I'll quickly pass this on to Helen and then to Chris. This is our friend covered barley. This is the barley that we're most familiar with. And then about 8,000 years or so ago, there was a domestication events that led then to the homozygous recessive condition at the nude locus. This is just showing 2 different color patterns. And so if you're not nude and you want to use the barley for our human food, then again, you're pearling that. So is it time, I would ask the question, to move to just shedding the hull in barley after 8,000 years? Because if barley simply didn't have a hull, it would really bring a lot of versatility to our crop. You would be able to potentially use the same variety for food, for feed, and for beer. This chickens are fed on a barley-rich diet here at Oregon State University, and this was one of our varieties, Buck Naked, and this was an ale brewed by a brewery in Portland. The loaves are thanks to our colleague, Andrew Ross here at Oregon State University. So why does barley have a hull? Why have we gotten ourselves boxed into this? Well, because a very important role that the hull plays in brewing during the lautering process. So the hull is a natural filtration medium. This is the malt of Thunder, a covered variety of ours with malts generated here at the OSU Malt House. And then this is just a range of naked barley malts. And these have, some rather interesting attributes. A number of the initial shots across the bow with naked barleys have confirmed the very high malt extract value. So as you look here in the first column, the first 4 rows are naked barleys, and just note those malt extracts. That's enough to salivate over compared to the covered control, which is Thunder down here in the bottom row. But the beta glucans get a bit chewy here, as you might see, but the buck is definitely up there. Scott Fisk has been able to bring that down in different tweaks to the malting process. But note that even as is, some of these double haploids here are able to achieve some reasonable beta-glucan profiles. And then as you look across the other malt profiles here, some of these things are approaching some reasonable specs in terms of enzyme activity and other characteristics. These are the pedigrees of this material. These were developed, the first 2 of these with our colleagues in Spain, Ignacio Romagosa and Luis Sistue, and then this is a line we developed in collaboration with the Virginia Tech program. So if you're going to be malting with naked barleys, what do you need to do? Well, if you're going to use all naked, then you may be wanting to go for a mash filter. So the mash filtration process allows you to completely bypass the lautering process, and or you may lauter and add filtration media such as rice hulls. So a few years ago, we just got to thinking, well, what about being able to use some gene editing in terms of achieving naked? And with this, then I'm going to send this over to Helen to talk about our experience with CRISPR in her hands. So all yours, Helen.
Cheers. Yeah. So just as Pat said, this came up in conversation of looking at the difference in malting traits specifically, and also disease phenotypes between these hulled and then the naked barleys as well. So the frustration for any genetic study with this is that there's a lot of diversity of genotypes. You can't just take a pool of naked barleys and then take a pool of hulled barleys, because you've got a lot of other genotype specific background effects as well. So the gene controlling this naked phenotype is a single gene NUD that is in this publication here that I'm showing you the figure of. So it makes it an ideal target for gene editing to generate these isogenic lines that we can then use to investigate the differences. So here, very nicely, just showing that panel A, you've got the hulled and then the hullless naked next to it as well. So if we go to the next slide, please, Pat. So that's great. We started this project and the gene editing, and then I think it was a couple of years into it, we got scooped as well. So another team realized that this was an ideal target for gene editing and published this really nice paper on what they did as well. But I'll continue with, the next slide. So here, I'm just showing you for anybody that's unfamiliar with CRISPR-Cas9 gene editing. So essentially you have a guide RNA target, which is highlighted in green here. And so we designed 2 targets that are on exon 1 and exon 2 of the NUD gene. And the way that CRISPR-Cas9 works is that you've got this PAM site, which is a 3 base pair, which is something GG, so it ends in a GG. And then there'll be a double-stranded break in between the guide RNA sequence and this PAM site indicated by my lovely graphical scissors there. So at the bottom of the slide, there's the coding sequence of NUD, and then I've highlighted where those 2 targets are. And so we were aiming for a cut to disrupt this gene, basically. So it's very close to the start of the coding sequence. And there I've just also highlighted that the way that we designed the targets was that the cut site you would aim for disrupts a restriction site. So therefore, when you have your transgenics, this makes it really easy to distinguish between what has been edited and what hasn't. So that if you amplify the coding sequence of NUD and then try and digest this in the cut or in the genotypes and the mutants where you have a successful cut, you won't get digestion of the product, and also this product will be a lot shorter as well. So that's in essence what we did. So really nice project that was very straightforward, and we managed to get very good mutant lines from these with successful phenotypes. Oh yeah, if we go to the next slide. Yeah, so here again we've got the wild type on the left hand side, and then our generated naked promise lines there. So really nice, super clear phenotypes. And so yeah, if we go to the next slide. And so perfect collaboration with Pat. So that was generated by the really great transformation team that we have here, the transgenic lines, and then the benefit of US over the UK. Well, who knows in future years, but the ability to do transgenic field trials as well. So we got these mutant lines certified transgene free. So to do this, we just ensured that we then had the disruption of NUD, but we didn't have any of the other CRISPR-Cas9 machinery present in the mutant lines. So therefore they are undistinguishable really as transgenic lines. So with that, we were able to get these, in essence, deregulated and into the field where we could start bulking to then go for downstream. Here we go, yeah, with the malting as well. So the idea then is that we've bulked up the seed so we would be able to malt our naked promise lines as well. I think with that, I'm now handing it back to you, Pat. Yeah, so here's it in the field as well. And so super interestingly, Pat was able to see a difference in phenotype. So on the left-hand side, we've got the wild type golden promise. And then on the right-hand side, we've got our naked promise line. So there's a difference in color as well that we were not expecting. So this then needs some follow-up because perhaps it's not so isogenic as we were hoping for. But in the design of the guide RNAs, that there, we double-checked that there weren't any off-target sort of sequences that we would hit, but this was prior to the release of the golden promise genome. So maybe going back now, we might find something else. And then interestingly, there was a difference in Threshability phenotype as well. So on the pictures of the seed at the bottom, you're not getting quite this free threshing phenotype as well with the mutants. So yeah, quite interesting, other unexpected effects that were going on. So yeah, back to you, Pan.
I'll just pass it directly on to Chris. Thank you, Helen.
Yeah, so this is where my work with on naked barley threshability comes in. You'll see in those past pictures, in those lots of seed, there was some variation on whether or not the hull actually remained attached. And this is what we, the propensity of the grain to actually shed the hull is what we're defining as threshability. And from this, we see that this recessive NUD mutation is required for removal of the hull, but it's not sufficient for complete removal of the hull. So we measure this trait on a 0 to 4 scale, which is what you see in this upper left-hand figure, with number 4 being all hulls removed and 0 being no hulls removed, as well as a few intermediates. There's not actually a lot of literature about threshability. What we could find going into this project is that the wide plump grains tend to show higher levels of threshability. Additionally, grains with low hull attachment have higher test weight on average. This is because the hull takes up about 10 to 13, or accounts for 10 to 13% of the grain weight by volume, or grain by weight, pardon. but it's not as dense as the grain, so we'll see higher levels of test weight in lots of seed that have less holes in them. We investigated these relationships in a population of 350 naked barley genotypes, which is what we see in the correlation matrix on the right. This is a Pearson correlation matrix with THR being the threshability, TW being test weight or weight by volume, then thin and plump being percentage of thin grain and percentage of plump grain for a given genotype. And we do see this great correlation between threshability and test weight, not so much with thin and plump though. Also, I really like this picture here in the lower left. This is courtesy of Dr. Hayes. Thank you very much for this picture. This is a very fine detail of one of the poor threshing naked promise seeds. And you can see that the hull isn't adhered to the underlying caryopsis like it would be in a naked or in a covered variety. But what we do see is this little point of attachment on this little bit of tissue where the grain was failed to be abscised, or where the hull failed to abscise from the grain. And that, I believe, is a key underlying mechanism in threshability. But that being said, we, going into this, did not know a whole lot about the genetic architecture, a whole lot about threshability as a whole. So if you can move to the next slide, Pat. Thank you very much. We started by conducting a genome-wide association study for threshability using those same 350 genotypes, as well as the Q plus K model for GWAS to account for population structure and kinship. As a note on population structure, there were 6 subpopulations identified in our 350 genotypes that we had to account for. And so what we see here from our Manhattan plot, our GWAS results, is 2 significant hits that cross the family-wise error rate threshold of 0.05, one on chromosome 2H and one on chromosome 3H. Investigating those markers a little bit closely, on this table on the bottom, we have there the marker name, the chromosome position data, as well as the effect size, frequency of the allele conferring high threshability, and then the phenotypic variance explained by this allele. The effects on either are not particularly large. Both of these together account for less than a 0.5 change in the threshability of a given genotype on a 0 to 4 scale, and neither one of them accounts for more than 10% of the phenotypic variance explained. So if we're talking in terms of an intentional marker-assisted selection strategy, these may not be that promising, but it does give us information and insight to the multigenic nature of threshability, which I think is interesting in and of itself to know that while the naked trait is conferred by a single gene, this full removal of the hull is definitely quantitative in nature. So if you could move to the next slide, Pat, thank you very much. So for traits controlled by many genes of small effect, genomic prediction and genomic selection can be a more effective means of achieving genetic gain if our goal is to select for threshability. We use the GBLUP procedure to predict threshability values in our population. So on the left here, you'll see a table. And it shows this model number 1 is the basic GBLUP model with no additional covariates, using information from our 350 genotypes in 9 environments across Wisconsin, New York, and Oregon. Using that procedure, we saw a predictive ability of 0.84. So our predicted threshability had a correlation with our observed threshability of 0.84, which is amazing. We were interested to see if we could further push that accuracy higher by, 1, including our identified markers from our GWAS analysis as additional covariates in this prediction model. And that is not what we saw. We saw no impact of these additional covariates. We also had these grain traits for percent thin, percent plump, and test weight in our Corvallis environments, in our Oregon locations. And we see that the addition of these as additional covariates did not increase our predictive ability. But that being said, 0.84 is still very high and very promising. Based on the high predictive ability, genomic selection has the potential to assist us in making genetic gain for threshability, particularly when we are trying to make rapid advancement in generations, and scoring threshability after harvest would reduce our efficiency. It's also my hope that we could use this strategy to pre-select covered barley if we were to introgress covered lines into our breeding pipeline. I would still need to test whether that is possible, but that's kind of a future avenue of research that I would be interested in. Thank you.
Thank you, Chris. Thank you, Helen. And I'll just wrap up this bit on naked barley, and then we'll just kind of whiz through the flavor part so that people can get back to their roasted barley tea or their beer or spirits or whatever the time of day permits. Just to point out that this whole, there are some stones on the path to being naked, and one of them is smut. And both the loose and the covered smuts are just nasty, and a lot of this production, at least now, is under organic conditions, and so they don't have the opportunity to treat seed. And so it's kind of a renaissance of resistance genetics and smuts is very much indicated. The other thing that is certainly worth considering is the retention of the embryo. So up in the upper right or the center panel here, you see barley seeds that really look just fine, but in fact, they are missing their embryos if you look carefully down at the distal end. And there is genetic variation for the retention of embryos, embryo retention in the upper right panel here. So two traits that definitely need to be worked on. My colleagues, Bridget Meintz and Christiana Vallejos here at Oregon State are tackling both of these issues. So this business of the embryo loss really can have an effect in terms of your field performance. In the bottom right panel, you'll see a plot that has very poor stand because of damage to embryos. And if you go into a malting regime with something that has lost a lot of embryos, that's definitely going to drive up your beta-glucan values. So, you know, we've got some prospects now for moving ahead to, I think, implementing facultative and naked together. And so Chris identified genomic selection as a strategy that we can certainly use for threshability in the naked barleys. On the facultative side, we're getting a good grip on many of the genes that are involved. And so I think it's quite promising that we can make significant progress, but I would just you know, being a real fan of phenotypic selection, I do believe that it's just essential to get these things out into field trials as quickly as possible. So now to just kind of wrap this up, just a little bit on flavor. And so flavor is very much in the eye of the beholder. It's in the nose, it's in the tongue. And ultimately, all of our barley products, I think, are merit exploration of flavor. So in the left panel here, you are seeing roasted barley teas. And this is an area of research in our group with Mariona Martinez-Subira, who's there as a postdoc. And so we're tackling this question of what do consumers think of roasted barley tea with a little bit of a twist to sort of put it into the Pacific Northwest craft profile. Here we see some research beers that we did in collaboration with our colleagues at Deschutes Brewery, and this then led to an extensive research pipeline in lots of contributions from so many colleagues here in fermentation science at OSU, Harmony Bettenhausen, currently at the Hartwick Center, and so forth. And then in breads, this is something that we just really haven't yet explored. What is happening in terms of food and food perception in terms of barley and sensory? So there are skeptics out there, as in the lower right panel. And so a colleague at a barley conference pointing out that, indeed that this quest for flavor might be academically proven, but it might not be process meaningful. And so we often find in our publications on flavor that reviewer number 2 raises their claws and their fangs, and they come out and really light into us about our explorations of flavor. But we have made some strides there and have developed a research pipeline that has been effective in identifying flavor in a limited panel of barleys, but we have extended it beyond the Golden Promise full pint panel that has been the focus of our initial research, and starting to move it into some more contemporary varieties. And so there's some literature there that I'd encourage you to look into. But most recently then, Sayre Chavez et al. Published in the JSC, and it's an open access paper, so you can pick this up, and it was mapping the barley genes in beer flavor. And so here we used the Oregon Promise population, where we crossed Golden Promise by full pint, went through micro malting, nano brewing of a malting population. So this was just absolutely heroic work at raw malting. Our colleagues there made these individual bottles of beer and then did the sensory on them. And then Harmony led the charge on the beer metabolomics. We coupled all of these data and then went into a QTL analysis in this biparental population to see what might be driving malting quality, metabolomic traits, and beer sensory. And lo and behold, we identified a number of QTLs, which you can explore in greater depth in the paper here. I just wanted to call your attention to 2 fascinating things that came out of this research. And one of them is on chromosome 3H, and this was SDW1 densa. And so, could morphology be having an impact on flavor? Well, you're looking at GA-related genes, and so GA is related to perhaps other downstream effects in terms of modification during the malting process. But the whopper that we have down here, well, let me just back up here. We also have ARI-E up here, so the other dwarfing gene, because we've got 2 dwarfing genes segregating in this population, could be SD1. And then down here, though, look at this whopper for multiple QTLs down here in the long arm of 5H. And lo and behold, who is this but MKK3, which is a well-known dormancy gene in barley. And, and so you ask the question, how does dormancy relate? These our barleys, our population, were not dormant. They were malted 2 years after harvest. But could there be a rheostat, a regulation of dormancy that could affect modification? And does modification in turn affect downstream sensory? So lots to explore here. And our colleague Casato has just recently published on CRISPR editing of SD2. So there's lots of opportunities out there to be exploring what is happening in terms of flavor. So how do we put all this together in terms of an FNF project? So FNF would be our acronym for facultative, naked, and flavorful. Well, it's kind of through a glass, but I think with some encouragement. And so as we look forward, I think that we can make some tremendous progress on a number of fronts with the sorts of tools that we've called on today, and with a re-galvanized barley community. So putting all this together, young barley researchers, disregard the advice of your elders. This is some advice that I got from the most renowned barley breeder of his time. And he just told me in regard to winter malting barley that it could not be done, and signed it and dated it. So make your crosses and create new germplasm and move all this forward into something new and exciting, and looking forward to this re-galvanized community and everything that they can achieve. And so thanks to all of these people out here, the Oregon Barley crew, all of the current members are listed here. Javier Hernandez has gone on to Universidad de los Lagos in Chile, and the rest of them are with us at this point. Helen's contributions you've heard. Lucia Gutierrez has led the charge on the genomics of naked traits. Maria Munoz was the lead on that Frontiers paper on facultative growth habit. Matt Moscow has just been there with us since the beginning on flavor and then subsequently with the Crispr project. So thanks to all of you, and time now for questions. I am going to unshare my screen so that then we can hopefully see a bit more.
That's a good idea, Pat. Then we can see everybody who's asking questions. So we've got a whole bunch of questions in the chat box, but I'm sure other people on the audience will also have questions they want to ask as well. So I'll start from the very beginning, and we'll ask them quite quickly. We've got about 10 minutes before 5 o'clock, so I might skip some of them, and particularly the ones that I asked, to let other people have a chance. Chris, have you seen a correlation between threshability and malting quality parameters?
That's a good question. And unfortunately, in my in this germplasm that I use, this 350 genotypes, the plots we're using are quite small, and it's not a conducive yield of grain to use directly anyway in malting trials. And so I don't have that kind of information on hand. I can say, just in general, what Pat showed about seeing higher extract levels in naked grains as opposed to covered grains. But I can't show any direct results from my experiments.
Okay. In addition to all the questions in the chat, I've got at least 4 people here with their hands up. So I'm going to flick between the chat box and those who have got their hands up, just the way I see them. So Isle, do you want to ask your question? Okay.
No, it was a mistake in the emoji. I was just clapping.
Okay, that's fine. Girma, do you have a question, Girma? I think we have some mistakes here in the hands up for questions. Now upon, do you have a question? Okay, I'm going to go back to the questions in the chat. So from Richard Pickering, is there any association between spike density and threshability? Because Golden Promise, you're correct in thinking, is quite a dense spike. It's got the ZO mutation in it.
This is going to have to be another situation where I say I don't really have that data. That is not something that I had considered looking at, and I'm actually going to add that to my notes because that is interesting to look at spike architecture and how that could be impacting these traits. But it's not something
it's one of the less severe alleles of ZO1. So from James to everybody. I'm not sure which James it is, but from James, maybe Brosnan, were your threshability numbers weighed to take account of kernel moisture levels? Because they found that the lower the moisture, the easier it threshes in the combine. So that's to Pat or Chris. Chris, you're getting them all. Pat just doesn't want to answer.
So our harvesting procedure, we harvest once. And so we're not differentiating between different
do you measure moisture after? Do you measure grain moisture levels after you've harvested?
It is part of our standard procedure in larger trials. Again, these experiments were done with 2 5-foot rows, and our equipment isn't really set up to measure moisture in that small of a amount in that small of a yield.
Okay.
Might call on our colleague Bridget Meintz, who's on I see her name here. She's kind of the lead on our organic naked research. She might want to comment on a couple of these themes that have come up. Bridget, are you there? Alrighty, so I'll just throw in a few things there, is that all of this is in process. So Bridget did just wrap up a series of brewing trials with naked barleys here in with the fermentation group across the street. And one of the I'd say something that popped out of it as an interesting barley was one of Kevin Smith's naked barleys that showed up with a that has poor threshability, but it came up with a nice, as I recall, a caramel malting profile. And the overall malt specs were not bad on it. So some hope there. And, you know, in terms of malting, if you have a bit of hull retention, that may drive down your extract a bit, but what the heck, you've got some built-in filtration value. So you may not need to use as many rice hulls if you're having to do that in a watering system.
Okay, I'm going to ask 3 questions at the same time because they're all kind of related. First of all, have you crossed naked promise to a hulled variety? And is the threshability problem due to background? And this is from Luke, who mentions that Golden Promise has zeal. The second question is similar. Do you have comparative micro-malting data on the Golden Promise and Naked Promise lines? And do you have germination data on the Golden Promise and Naked Promise lines?
See, I'll tackle those, and then Chris, if you could weigh in a bit. So we did make some crosses on the Naked Promise with various covered barleys. And it's really vicious to be trying to cross to something that short under field conditions, because that's the only place that we had it going. Don't know the status of that F1 seed, but we passed that on to Chris, who may give a thumbs up if any of it's germinated or not. We have also shared that Naked Promise seed with Molson Coors, who is quite keen on developing naked malting semi-dwarf barleys. And so they knew the Crispr origin of it. They knew the threshability issues, but they were brave enough to say, hey, maybe we could move that into their spring covered program using Chris's genomic selection strategy to select for improved threshability. So no malting data yet on those, but you know, that will hopefully be forthcoming with 2022 harvest.
Okay, so Dave Griggs makes a comment which is probably relevant to this. He's going to talk about Maris Otter. And what David says is, of course, that barley variety flavour does persist into final products, beer and spirit. And that's one of the reasons that Maris Otter is entering its 57th harvest this year. So you're obviously onto something. Luke Ramsey asks, what's the impact of naked on flavour?
Oh gosh, let's see, flavor trials. We will assess that in a preliminary way with the work that Bridget's doing now with the fermentation people, because the control was Copeland in both lauter and mash filter, and there were 3 naked barley malts in that experiment, as I recall. So data coming.
Okay.
Hey, sorry. Do you want me to comment on that?
Yeah, of course.
Please.
I've just got to your comment here.
Yeah,
Bridget,
sorry.
please.
I was typing and then realized I could have just unmuted myself. So for the brewing study that we just did, we did have 3 naked barleys compared with Copeland, and we used both a lauter tun and mash filter. And actually we found that we clogged the mash filter 3 times, so that perhaps may not be the answer to brewing with naked barley. But the lauter tun with the rice hulls performed successfully every time. So while rice hulls may not be the desired addition, we do have future studies coming up actually doing a combination covered and naked malt. But in terms of
We've lost you.
Oh, right at the cusp of excitement.
Yeah, there's a punchline.
Anyways, great stuff is coming. Stay tuned. That's the message. And anyone else doing this kind of work, please let us know. Get in touch with Bridget and the rest of us, and let us know where you're headed, because the more of us collaborating on this, the more progress we'll make.
So, Maria, would you like to ask the question?
Yeah, thanks. A very interesting talk. I was wondering, I mean, you mentioned in the beginning that you're aiming at, if I understood you correctly, separating the photoperiod sensitivity from the vernalization requirement. And from this, I was wondering also in a larger context, to what extent is pleiotropy of the genes that you're targeting or manipulating relevant for you? I mean, for the photoperiod sensitivity and vernalization, isn't it that to some extent these are controlled by the same genes, so difficult to separate really? Or do you think you can really separate these responses genetically?
Well, for sure, there's a continuum of phenotype across these things, and so we can break things out fairly discreetly in terms of the vern loci. So those would be the classically described vern 1, vern 2, vern 3, and then in terms of photoperiod with PPDH1 and PPDH2. But then when you get into a whole plant phenotype situation, be it in a controlled environment or be it in a field situation, of course, these things are interacting and confounding one another. But really the most challenging one for us to phenotype is the PPDH2 deletion or the null allele, the short day sensitivity, because to do that in a controlled environment context, you'd want to have short days for an extended period of time in a growth chamber, and ours always break down before you can ever get to the end of the experiment. So that's really one where we would want to, to have a marker diagnostic for that. In terms of pleiotropic effects, I expect that they are going to be important and huge because even this most recent issue of Science was describing some of the yield component impacts in wheat with growth with genes that are involved in flowering and growth and development. And so I think that we're probably going to see these. And so that may be one of the factors that's accounting for performance in our variety Lightning, which does very well in fall planted trials. That's where we selected it. But in a spring planted trial, it's kind of in the middle of the pack. It's not out there beating the best of the springs. We haven't done the detailed studies that are required in terms of phenology, morphology, and yield components to understand what's going on. But it's certainly not going to be as easy as it looks in the seminar.
Yeah, but you're optimistic then that you can break the genetic correlations between some of these pleiotropic traits.
Well, one, if you're going to be a barley breeder, you have to be optimistic.
Okay, yes.
Otherwise, it's going to be really rough out there. But I do have hope. And the facultative bit is just an important little item to plug, is that over the years, I've had many very experienced breeders just say, if you're going to have maximum cold tolerance, you have to have a vernalization requirement. And I think that we've shown that is not necessarily the case, that the association of vernalization sensitivity with low temperature tolerance is really a correlation rather than a causation kind of event.
Okay, yeah, thanks a lot.
Pat, Helen, Chris, I'm going to take two more questions and then call it a day, if that's okay with you. The first one's from Tom Blake. You have to unmute, Tom.
Sorry, that was probably a technical error from his
Yeah,
wife.
hey, Robbie. But I would like to make one comment. Paul Schwartz, postdoc, gave a seminar at an ASBC meeting two years ago that showed that about 75% of Fusarium hyphae wound up remaining in the hull after the malting process. And by getting rid of the hull, maybe you could get rid of 75% of the problem of Fusarium. And I asked the question to the crowd, so does that mean we should go to naked malting barleys? To which all of the industry guys turned around and gave me very nasty stares.
Thanks for that, Tom. Ayal, you still get your hand up.
Sorry, I have an update on that. Actually, Kevin Smith has a graduate student working on...
I don't know why. Sorry. We'll put it off.
Oh, again,
Kevin Smith has a graduate student working on looking at dawn accumulation in the hull of 250 naked barleys post-threshing, looking at grain accumulation and hull accumulation. So stay tuned for that.
Yeah, and just to follow up on that point as well. So that was what we were hoping that the Crispr the lines would be really useful for as well is doing these really clean experiments with these near isogenic lines as well. So that's why, again, the difference in phenotypes was also frustrating. But yeah, I'd love to do those sort of experiments as well.
So we've had all the questions. I just got a quick one on that point, Helen. How many independent transgenics did you look at in your experiments?
Yeah,
So-
so we do have multiple transgenics. So we have a lot more than just those 2 lines that we focused on. So yeah, we followed up with those 2 independent transgenic lines and just ensured that those were transgene free and just had the editing. So yeah, good point. There's probably a lot more lines to go back to as well. We didn't see any phenotypic differences when we grew these in the glass house, so it was really putting them in the field that made the difference.
Okay. Listen, just after 5:05, I think we've had a really good afternoon. I would just like to thank everybody again for the presentations, for the questions, for attending. And, you know, in a month's time, we have somebody from Australia speaking, Kenton Porker. He's an agronomist. If you can join us, please do. I think it'll be very difficult for the people in the US to join live, but remember, these things are recorded and you can go online and listen to them if you want to afterwards. So thanks again, just to Pat and Helen and Chris and everybody for joining us. So see you in a fortnight or in a month's time. Bye-bye.
Paul.
Yay.
See you later.
Robbie, we look forward to cycling with you out here.